Okay, good morning all. And now let us continue the chapter nucleic acid chemistry. First of all, what we have discussed in the last class, we have seen the structure. We have discussed about the structures of nitrogen bases, purines and pyrimidines, and the structure of sugars which participate in the structure of nucleic acids. And we all know that purines or nitrogen bases are of two types, purines and pyrimidines. Purines are adenine and guanine. These are formed by the structure is formed by the fusion of a pyrimidine ring with the imidazole ring. And here the atoms, there are 4 nitrogens and 4 carbons are there, and they have given the numbers like in anti-clockwise direction, 1 2 3 4 5 like that till 7. And adenine and guanine are the purines, which you can see in the structure of both DNA and RNA. And coming to the pyrimidines are thymine, cytosine and uracil. And here these are only 6 membered ring structures. Out of that 4 are nitrogens and 2 are carbons, and they have been allotted the numbers in clockwise direction. Now here cytosine is a pyrimidine which is present in both DNA and RNA, whereas thymine is present only in the structure of DNA and uracil in RNA. And coming to the sugars which are present, based on the sugar only nucleic acids, or like we can call it as, if the sugar is ribose, that is ribonucleic acid, like ribonucleotide, and if the sugar is deoxyribose, so that we can call it as deoxyribonucleotide. Now here this is the these are the 2 pentose sugars, ribose. Here the which exist in furanose form. There are 5 carbons are there here, and here this is the structure of ribose, and if one oxygen is less at second carbon, that is to form like deoxyribose. Ribose is the sugar present in RNA, whereas the deoxyribose is the sugar of which is present in the DNA. Okay. Now here now coming to the nucleosides. So how a nucleoside is formed? Okay, whenever a sugar, okay, the sugar may be ribose or deoxyribose, whenever it is attached to a nitrogen base, a nucleoside is formed. Sugar is attached to a nitrogen base, a nucleoside is formed. The nitrogen base may be purine or pyrimidine. Suppose if the pentose sugar is attached to a purine sugar, the linkage is the so formed linkage, we can call it as beta N glycosidic bond. That bond joins N9 of the purine ring to the C1 of the sugar if it is a purine nucleoside. And if it is a pyrimidine nucleoside, the beta N glycosidic bond joins N1 of the pyrimidine to the C1 of the pentose sugar. Okay, so in order to differentiate the atoms of the sugar and nitrogen bases, the atoms of the like sugars have been given the prime numbers like 1 prime, 2 prime, 3 prime, 4 prime and 5 prime, and the atoms of the pure bases like purine bases or pyrimidine bases, they are given with the cardinal numbers like 1 2 3 4 5 etc. Okay, but atoms of the to differentiate the atoms of between these two, the atoms of the sugars, pentose sugars, have been given the allotted the like prime numbers 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. Now coming to the like this purine nucleosides and pyrimidine nucleosides are formed. Now coming to the formation of a nucleotide. Already we know that nucleic acids are polymers of nucleotides, so they are made up of nucleotide. So what is a nucleotide? So whenever a phosphate group is attached to a nucleoside to form a nucleotide. So already nucleoside means where the sugar is attached to the nitrogen base by beta and glycosidic bond. To this, whenever these are phosphorylated to form nucleotide. So that means where the phosphate group has been attached. Is it attached to nitrogen base or sugar? So always the OH groups which are present in the structure of sugar, they are esterified with the phosphoric acid. Okay, now here in the nucleoside formation first, sugar is attached to the base by beta N glycosidic bond. Now here in the formation of a nucleotide, the phosphates have been attached to OH groups which are present in the structure of ribose sugars. Now here like nucleotides means they are nothing but phosphorylated nucleosides. Now the composition of nucleotide, already we know that composed of base sugar and phosphate. So sugar, whenever sugar, if it is attached to a base to form a nucleoside, and if the phosphate is attached to the sugar to form nucleotide. Okay, listen, sugars have been attached to base to form nucleoside by beta and glycosidic bond, and the OH groups of the sugars are esterified, has been esterified with the phosphoric acid to form a nucleotide. Now nucleotides are defined as nucleotides are nucleosides to which phosphoric acid has been attached to a sugar molecule by esterification at OH group. Suppose you see, if the sugar is like ribose sugar, the OH groups are present. If the sugar is a if the sugar is a ribose sugar, the OH groups are present at 3 prime, 4 prime and 5 prime. Suppose if the sugar is a deoxyribose, the OH groups are present only at the 3 prime and 5 prime. See here, the phosphate groups may be attached to the sugars at the OH groups. In case of ribonucleotides, so the OH groups, OH groups are present at the 2 prime, 3 prime and 5 prime. In case of deoxyribose, OH groups are present at 3 prime and 5 prime. So that means the possible sites for esterification of ribonucleotides is maybe phosphate groups may be attached to, may be esterified with the 2 prime carbon, 3 prime or 5 prime. In case of deoxyribonucleotides, the possible sites for esterification with the phosphoric acid is either 3 prime or 5 prime. Now here, so like that nucleotides are formed. So depending upon the type of the sugars present, nucleotides are of two types, ribonucleotides and deoxyribonucleotides. If the sugar is ribose, it is ribonucleotide. If the sugar is deoxyribose, to form deoxyribonucleotide. In case of ribonucleosides, already we have discussed the possible positions for phosphate esterifications are 2 prime, 3 prime and 5 prime, because where the OH groups are present. In case of deoxynucleosides, there are only two sites for two possible positions for esterification, that is 3 prime and 5 prime. Now here nomenclature, then coming to the nomenclature of nucleotides. So nucleoside, if it is phosphorylated on 3 prime carbon, it is termed as nucleoside monophosphate. Okay, see here the possible sites are I've already told you. In case of ribonucleosides, 2 prime, 3 prime and 5 prime. In case of deoxyribonucleotides, there are only 3 prime and 5 prime. Suppose if the phosphorylation done on 3 prime hydroxycarbon, the so formed nucleotide is termed as nucleoside 3 prime monophosphate. If the nucleoside is phosphorylated on 5 prime hydroxyl group on 5 prime carbons, they are termed as nucleoside 5 prime monophosphate. Okay, the possible sites in deoxyribonucleotides are for esterification with the phosphoric acid are either 3 prime or 5 prime. If the phosphate group has been attached at the 3 prime hydroxyl group to that is termed as nucleoside 3 prime monophosphate. If the phosphorylation done on 5 prime hydroxyl group, they are termed as nucleoside 5 prime monophosphate. But most commonly, usually the esterification is done with the 5 prime hydroxyl group. Okay, usually in most of the time in the formation of the nucleotides, the phosphoric acid is esterified with the 5 prime carbon of the pentose sugars. So that's what we no need to mention every time it is nucleoside 5 prime monophosphate. Simply suppose if it is, so that's what the 5 prime is omitted. Suppose you take adenine. Adenine is the base. Okay, when it is bind with the sugar ribose to form a nucleoside adenosine. Whenever the 5 prime carbon of the adenosine is esterified with the phosphoric acid, the so formed nucleotide we can call it as adenosine monophosphate. Okay, I repeat, adenine plus ribose to form adenosine. Adenosine is a nucleoside. Whenever it is phosphorylated, I already told you, usually phosphorylation done at the 5 prime hydroxy group of the sugar, of the pentose sugar to form a nucleotide. So adenosine on phosphorylation to form AMP. Okay, to form a nucleotide which is known as which is abbreviated as AMP. AMP stands for adenosine monophosphate. Here we no need to mention it is 5' AMP, because AMP that indicates that there is a 5' carbon has been esterified with the phosphate. So when we have to mention 3' means, suppose if the esterification is done at the 3' hydroxyl group, then we have to mention it is 3' AMP. So without mentioning that, simply we write AMP, ADP or CMP, means that denotes that the esterification is done on the fifth prime carbon of the ribose sugar. Now here suppose already one phosphate has been attached to the suppose you in case of AMP, already one phosphate hydroxyl group at the fifth prime of the ribose sugar is esterified with the phosphate to form a monophosphate nucleotide, that is adenosine monophosphate. If additional phosphate is attached to the existing phosphate, already one is there, if additional second one is attached to the existing one to form nucleoside diphosphate. This nucleoside diphosphate on further phosphorylation to form nucleoside triphosphate. Okay, so nucleoside triphosphates are usually abbreviated as NTP, that is nucleoside triphosphate if the sugar is ribose, or if the sugar is deoxyribose, that we can abbreviate as deoxy nucleoside triphosphate. Okay, now nucleoside diphosphates are how many? In nucleoside diphosphates, in between two phosphates there is one high energy acidic anhydride bond is there, one high energy bond is there. In case of triphosphates, nucleotide triphosphates, there are two high energy bonds are there. Okay, this in case of diphosphates, there are one high energy bond is there between the two phosphates, and in case of triphosphates, there are two high energy bonds are there in between the among the three phosphates. Now here you know that high energy bonds which on hydrolysis, each bond on hydrolysis, that is called squiggle bond, or you can call it as acid anhydride bonds, which on hydrolysis yields more than 7 kilocalories of 7.6 kilocalories of energy. Now here coming to the structure, let us see the structure of AMP. Okay, see here AMP is one adenine nucleotide, which is AMP stands for adenosine monophosphate. First see the structure of adenine. Okay, next one here in adenosine monophosphate, the sugar is ribose. So ribose is attached to adenine. The linkage is N9 of the adenine is linked to 1 prime. See the numbers, prime numbers. 1 prime of the C1 of the ribose is attached to N9 of the adenine to form a nucleoside. To this nucleoside, the phosphate OH group which is present at the 5' carbon of the ribose is esterified with the phosphate to form adenosine monophosphate. Suppose to this structure only, if one more phosphate is attached to the existing phosphate to form adenosine diphosphate, on further phosphorylation to form the universal currency of energy, that is adenosine triphosphate. And let us see the structure of this one. This is the pyrimidine nucleotide, that is TMP, thymidine monophosphate. Here the first sugar is ribose. Here the ribose is attached to the nitrogen base, that is thymine, by the linkage by like beta and glycosidic linkage, which connects N1 of the thymine to the 1 prime. Okay, 1 prime C1, 1 prime of the ribose sugar. Now here the OH group of this OH group which lies on the 5 prime of the ribose sugar is esterified with the 1 phosphoric acid here to form a structure known as mononucleotide, to form a mononucleotide, that is thymidine monophosphate. Okay, now here let us see the bases, principal bases, nucleosides and the nucleotide formation. For example, first let us discuss about the nucleotides which are present in RNA. Okay, here adenine, and here the adenine nucleoside is, here the sugar is ribose to form adenosine. The ribonucleotide, okay, 5' monophosphate is adenosine 5' monophosphate, or you can also, that is AMP, or also you can call it as adenylate. And guanine, its nucleoside is guanosine along with the ribose, and guanosine is phosphorylated to form guanosine 5' monophosphate or guanylinate. Its abbreviation is GMP. GMP stands for guanosine 5' monophosphate, or you can call it as guanylinate. Like cytosine. Cytosine with the ribose to form cytidine, its nucleoside, and cytidine with the phosphate to form cytidine 5' monophosphate. Uracil plus ribose to form uridine, and uridine is phosphorylated to uridine 5' monophosphate, that is UMP. And coming to the deoxyribonucleosides, adenine with the sugar deoxyribose to form deoxyadenosine, and it is phosphorylated to deoxyadenosine monophosphate, that is DAMP. Guanine with the deoxyribose to form its nucleoside, that is deoxyguanosine, which is phosphorylated to form a nucleotide, that is deoxyguanosine 5' monophosphate, so that is abbreviated as DGMP. And the cytosine with the deoxyribose to form deoxycytidine to form deoxycytidine 5' monophosphate, or DCMP. Next, thymine. Thymine is present only in the structure of DNA, which is along with the sugar deoxyribose to form deoxythymidine, and this deoxythymidine is phosphorylated at the 5' hydroxyl group to form DTMP, that is deoxythymidine 5' monophosphates. Okay, now here ribonucleotides, like these are the examples. So adenosine, which on phosphorylation first to form at the 5' hydroxyl group, if it is OH group is esterified with the phosphoric acid, first to form AMP, which on further phosphorylation to form ADP, adenosine diphosphate, on further phosphorylation to form ATP. One high energy bond is there in ADP and two high energy bonds are there in ATP. Like that, coming to the guanine with the ribose to form guanosine and its nucleotides, or if one phosphate is attached at the 5' hydroxyl group to form guanosine monophosphate, on further phosphorylation to form GDP, which on further phosphorylation to form GTP, that is guanosine triphosphate. Coming to the cytosine with the ribose to form cytidine, and cytidine on phosphorylation to form CMP, that is cytidine monophosphate. And cytidine monophosphate means here no need to mention 5', that indicates that their 5' hydroxyl group have been esterified with the phosphoric acid. And the further phosphorylation, addition of the one more phosphate group, the existing one, to form CDP, and on further phosphorylation to the existing one to form cytidine triphosphate. Then coming to uracil plus ribose to form a nucleoside, that is uridine. Uridine on phosphorylation to form its nucleotides UMP, UDP and UTP. UDP is uridine diphosphate and UTP is uridine triphosphate. So these are the ribonucleotides. And coming to the deoxyribonucleotides, already we have discussed. Adenine, if the sugar is deoxyribose to form a nucleoside, that is deoxyadenosine. Deoxyadenosine on esterification at the 5', if the ribose, deoxyribose, if it is esterified at the 5' hydroxyl group with the phosphoric acid to form deoxy-AMP, that is deoxyadenosine monophosphate, on further phosphorylation to form DADP and DATP. Like that, cytosine plus deoxyribose to form it's a nucleoside, that is deoxycytidine. On further phosphorylation, addition of phosphate groups at the 5' to form DCMP, DCDP and DCTP. Now thymine with the deoxyribose to form a nucleoside, that is deoxythymidine. This on phosphorylation to form its nucleotides, its mono, di and trinucleotides, or DTMP, DTP and TTP. Sorry, here it is TTP. And now here the last one is guanine, and here the guanine plus deoxyribose here to form deoxyguanosine. It's a nucleoside, and which on phosphorylation to form DGMP, sorry here it is written wrong, and which on phosphorylation to form DGDP and DGTP. Okay, you make the correction here. GMP nucleotides or GDP, DGDP and DGTP. Now here the in the above that also there is a correction is there, DTMP, DTDP, and the last one is deoxy TTP, that is thymidine triphosphate. These are the deoxyribonucleotides. Now I think you got, now you understood so far, like nucleotides means here made up of a base, ribose and phosphate. The base may be a purine or pyrimidine, and the sugar may be a ribose or deoxyribose. First these two joined by beta and glycosidic bond to form a nucleoside, and to the nucleoside, the OH groups of the sugars, if they are esterified with the phosphoric acid to form nucleotide. If one phosphate is attached, mononucleotide. If two, dinucleotides, and if three, trinucleotides. Now here coming to the unusual nucleotides. Okay, in the structure of RNAs, there are some unusual bases also you can found, that is pseudouridine. Okay, pseudouridine is present in tRNA, in the cloverleaf structure of the tRNA. Here pseudouridine, how it is being formed means, here ribose is attached to uracil at the 5th carbon. Okay, usually normally where it is attached to, in case of uracil as it is a pyrimidine, the attachment should be N1 of the uracil may be attached to C1 of the sugar. But here in this case, the 5th carbon, not the N1, the 5th carbon of the uracil is attached to first carbon of the sugar. So there is a carbon-carbon linkage here instead of nitrogen-carbon linkage. So it's an unusual nucleotide. By the attachment of this, nucleoside is formed, which is known as nucleoside is formed, which is known as pseudouridine, which is present in the structure of tRNA. Then the other one is dihydrouridine. Okay, this dihydrouridine is also present in the cloverleaf structure of tRNA. It is also found in cloverleaf structure of tRNA, and it is also a pyrimidine nucleoside. Here also there are two hydrogen atoms are attached to uridine. Okay, two hydrogens are attached to uridine here in the structure of dihydrouridine, and in the pseudouridine, the 5th carbon of the uracil has been attached to C1 of the ribose instead of there is N1 and C1 linkage, there is carbon-carbon linkage. So these are unusual nucleotides by name pseudouridine and dihydrouridine. Both you can see in the structure of tRNA. Then coming to cyclic nucleotides. Okay, till now we have discussed about the unusual nucleotides and then regular normal nucleotides, which you can see in the structure of structure of DNA and RNA. Then coming to cyclic nucleotides. So what are these cyclic nucleotides? So here the single phosphate group is esterified with the 2 hydroxyl groups. Okay, cyclic nucleotides are formed when OH groups of 3' and 5', OH groups of both 3' and 5' are esterified with a single phosphate. That would result in the formation of cyclic nucleotides. Examples are cyclic AMP, CAMP, and the next one is cyclic GMP, that is CGMP. Okay, now here the cyclic nucleotides are, so here normally in the nucleotides either 3' or 5' or hydroxyl groups may be esterified with the phosphoric acid. But in case of cyclic nucleotides, both the OH groups of both 3' as well as 5' both will esterify with the single phosphate group. So that would result in the formation of a cyclic structure, which is known as cyclic nucleotides. Okay, so examples of the cyclic nucleotides are CAMP, CGMP. Now CAMP is stands for adenosine 3' 5' monophosphate. Okay, here it's an adenosine nucleoside. So single phosphate has been esterified with the 3' hydroxyl and 5' hydroxyl. So chemically it is known as adenosine 3' 5' monophosphate. And this CAMP is synthesized from ATP by the membrane bound enzyme adenylate cyclase. And the CAMP is degraded by the enzyme phosphodiesterase to form 5' AMP. Then coming to CGMP. Okay, then CGMP is formed from GTP by the enzyme membrane bound enzyme guanylynated cyclase. Now this guanylynated cyclase is stimulated by calcium ions, and CGMP, it acts as a second messenger. Okay, now examples of cyclic nucleotides are CAMP and CGMP. CAMP is derived from ATP by the enzyme adenylate cyclase, and CGMP is formed from GTP by the enzyme guanylate cyclase. So both the CAMP and CGMP acts as secondary messengers for hormones. And these are examples, both these two are examples of cyclic nucleotides. Here only one phosphate is esterified with the 2 hydroxyl groups, which are which present on 3' and 5'. Then coming to the importance, biological importance of nucleotides. Now let us discuss about the first one, adenosine nucleotides. Adenosine nucleotides examples are AMP, already we have discussed AMP, ADP, ATP and cyclic AMP. All these are adenosine nucleotides. And first coming to ATP, already we know that it is a storehouse of energy, universal energy currency. And AMP is phosphorylated to form ADP, and coupling of ADP with inorganic phosphate to form ATP. Now this ATP which is synthesized by two ways, majority is by oxidative phosphorylation, okay, and the second one is oxidative phosphorylation is here where oxidation is coupled with the phosphorylation in electron transport chain. ATP is formed, and even ATP is formed directly from the substrates by substrate level phosphorylation. Okay, now here this ATP is formed by oxidative phosphorylation or by substrate level phosphorylation. Here substrate level phosphorylation means what? Here substrates are strong enough, the energy which is there in the substrate is strong enough to couple ADP with inorganic phosphate to form ATP. Okay, now here in the structure of ATP, two high energy bonds are there and two acid anhydride bonds are there in the structure of ATP. Each one on hydrolysis yields more than 7.6 calories of energy. So you know the reactions. ATP, when it is hydrolyzed to ADP, in hydrolysis of ATP it is hydrolyzed to ADP and PI with the release of 7.6 calories of energy, and ADP is further hydrolyzed to AMP and inorganic phosphate, again the release of 7.6 kilocalories of energy. Okay, look at the structure of ATP. Here adenine and the sugar is ribose. First adenosine is formed. The 5' hydroxyl group is esterified with the 3 phosphate groups. So in between the 3 phosphates there are 2 high energy bonds are there. Each one in hydrolysis yields 7.6 kilocalories of energy. Now here, so let us discuss about the functions of ATP. It's a main source of energy. So the energy which is trapped in ATP is utilized for all the endergonic reactions to take place in the body. Endergonic reactions means those reactions requires energy, and that energy is utilized for muscle contraction, and it provides energy for transmission of the nerve impulse and transport of the nutrients across the membrane. For example, you take active transport. Active transport means what? Here it is a solute moves against the concentration gradient and it is a carrier mediated energy requiring process. That energy comes from hydrolysis of ATP. So ATP provides energy for active transport. This transport helps in transfer of nutrients across the membranes, and even ATP provides energy for motility of the spermatozoa. So ATP as it contains 2 acid anhydride bonds which on hydrolysis yields provides energy, so that may be utilized for endergonic reactions, for muscle contraction, so the transmission of the nerve impulse, transport of the nutrients across the membrane, and for the motility of the spermatozoa. Next, ATP, even it is required for the formation of the SAM. What is SAM? SAM stands for S-adenosyl methionine, which is active methionine, and it is formed from the methionine where the phosphate donor is the ATP. Now here active methionine SAM, and SAM is the methyl donor in all transmethylation reactions. Now ATP, it donates phosphates for all kinase reactions. Okay, in the metabolisms, if you remember, wherever the kinase reactions are there, okay, wherever the kinase reactions, phosphate is required. The phosphate donor is ATP. For example, you take glycolysis. The first reaction is catalyzed glucose to glucose 6-phosphate by the enzyme hexokinase or glucokinase, where one ATP is hydrolyzed to ADP and PA. Now here ATP is hydrolyzed to ADP. Now here the next one, ATP is required for the formation of the PAPS, that is active sulfur. PAPS is phosphoadenosine phosphosulfate. So ATP is required for the formation of active methionine, and ATP is also required for the formation of the active sulfur, that is phosphoadenosine phosphosulfate, which is the sulfur donor in all the sulfur requiring reactions. And here this ATP, as it is ATP is required for all the biosynthetic reactions. For example, for the formation of the phosphocreatine from the creatine, and for the synthesis of fatty acids from acetyl-CoA, ATP is required. And here ATP is required, like all these biosynthetic reactions. formation of the glutamine from glutamate, and for the synthesis of glucose from pyruvic acid by gluconeogenesis, and for the formation of the proteins from amino acids. So for all the biosynthetic reactions, energy is required, which is provided by ATP. And these are all the examples. Then coming to PAPS. So PAPS is that is phosphoadenosine phosphosulfate. Okay, PAPS stands for phosphoadenosine phosphosulfate, which is active sulfate. It is formed from ATP and sulfate. Now it is a sulfur donor. The PAPS is a sulfur donor for the biosynthesis of mucopolysaccharides like heparin, chondroitin sulfate. These two are the sulfated mucopolysaccharides. Here the sulfur donor, sulfur comes from the PAPS. Okay, PAPS is synthesized from ATP and sulfate. So it is a PAPS is a sulfur donor required for the it is required for the biosynthesis of, it provides sulfur for the biosynthesis of mucopolysaccharides like heparin, chondroitin sulfate, and even sulfolipids. Now here even the PAPS helps in conjugation of the indole and skatole compounds to the ethereal sulfates. Okay, next one is here coming to the another adenine nucleotide, that is ADP. ADP is adenine dinucleotide, and here it is a primary phosphate acceptor in oxidative phosphorylation. In oxidative phosphorylation, what happens? ADP coupled with the inorganic phosphate to form ADP. So it is the primary phosphate acceptor. So that means ADP has a role in cellular respiration, and it has a role in muscle contraction, and ADP activates the enzyme glutamate dehydrogenase. Okay, these are all the functions of ADP. Mainly it is a primary phosphate acceptor in oxidative phosphorylation, so which plays an important role in cellular respiration, and it has a role in muscle contraction, and it activates the enzyme like glutamate dehydrogenase. Then coming to AMP. AMP is adenosine monophosphate. Adenosine monophosphate is usually it is produced by the degradation of the cyclic AMP by the phosphodiesterase. Okay, AMP means first it is the like ATP is hydrolyzed to ADP. ADP is on further hydrolysis to form AMP. Not only that, it is also produced by the degradation of cyclic AMP by phosphodiesterase. And here it is activator of the several enzymes. That is, in glycolysis it stimulates the enzyme phosphofructokinase. And in resting muscle, AMP is formed from ADP by the enzyme adenylate kinase or myokinase. Okay, see here in the resting muscle, AMP is formed from ADP by the enzyme myokinase. There what happens? Two molecules of ADP to form, two molecules of ADP means two adenosine diphosphates to form one ATP, adenosine triphosphate, plus one molecule of AMP. So that AMP which is released in the muscle stimulates the enzyme glycogen phosphorylase B. The glycogen phosphorylase B is stimulated means which will stimulates glycogenolysis. So that increases the glycogen breakdown in muscle. I repeat, in resting muscle, AMP is formed from ADP by the enzyme myokinase. The reaction is here. Two molecules of ADP gives rise to ATP plus AMP. The AMP which is released in the muscle then stimulates the enzyme glycogen phosphorylase B. That enzyme in turn increases the glycogenolysis, breakdown of the glycogen in muscles. Now here coming to the cyclic AMP. 3' 5, it is already cyclic nucleotide. Okay, it is chemically known as 3' 5' adenosine monophosphate, and it is formed from ATP by the membrane bound enzyme adenylate cyclase, and this adenylate cyclase activity is under the control of hormones, and this cyclic AMP is degraded to 5' AMP by the enzyme phosphodiesterase. Now here the cyclic AMP acts as a second messenger for hormones. Example, FSH, MSH, TSH, calcitonin, vasopressin. For all these hormone action, this cyclic AMP to exhibit their action, cyclic AMP acts as second messenger. This cyclic AMP in turn derived from ATP by the enzyme adenylate cyclase. Now here it increases glycogenolysis, cyclic AMP, and it increases acid secretion from gastric mucosa, and it also decreases the aggregation of the platelets. Okay, the functions of cyclic AMP is it acts as a second messenger. You can write the examples, hormone examples: FSH, MSH, TSH, calcitonin, vasopressin, and which stimulates glycogenolysis, and it stimulates the secretion of the acid from the gastric mucosa, and it prevents the aggregation of the platelets. Then this is the structural difference between AMP and cyclic AMP. Okay, here in the first structure, adenine to ribose to form adenosine. At the 5' hydroxyl group, it is phosphorylated with the hydroxyl group is esterified with the phosphoric acid to form adenosine monophosphate. And look at the picture, cyclic AMP. Here what happens? The OH groups which are present at the 3' and 5' both have been esterified with a single phosphoric acid to form a cyclic structure that is known as cyclic AMP. Okay, that is the difference between the AMP and cyclic AMP. And we have already discussed the functions of AMP and cyclic AMP functions separately. Then coming to the nucleotides of uracil. Uracil nucleotides are UMP, UDP and UTP. UDP glucose is the glucose donor for glycogen synthesis. If you remember the steps of glycogenesis, the active glucose, that is UDP glucose, is formed. Okay, that is the glucose donor for glycogen synthesis. Okay, what is UDP glucose? That is nothing but a nucleotide of uracil. Now here UDP sugar derivatives participate in sugar epimerization. So that means which help in the interconversion of glucose 1-phosphate to galactose 1-phosphate. Here, okay, see here, and the next one, UDP galactose, UDP glucuronic acid and UDP N-acetyl galactosamine are the sugar donors. So sugars mainly attached to nucleotides, and to form the active sugars, they may be participating, they may act as sugar donors. Examples are UDP glucose, UDP galactose, UDP glucuronic acid and UDP N-acetyl galactosamine. And coming to UDP glucuronic acid, already we know that it's a conjugating agent, or it helps in the conjugation of the bilirubins. Okay, and it helps in the conjugation of the drugs such as aspirin. And even UDP glucuronate, it has a role in metabolism of detoxification. Okay, if you remember the steps of conjugation of the bilirubin, there the conjugating agent is UDP glucuronate. UDP glucuronate means glucuronate is attached to UDP. UDP is a nucleotide. Next, nucleotides of cytosine. Examples are CMP, CDP and CTP. Cytidine monophosphate, cytidine diphosphate and cytidine triphosphate. Now CTP may be required for the biosynthesis of glycoproteins and phosphoglycerides. CTP, it is required for the biosynthesis of glycoproteins and phosphoglycerides. And CDP choline, okay, CDP choline is attached to the nucleotide CDP to form CDP choline, which help in the synthesis of the sphingosines and sphingomyelins. So these are all the physiological importance of the nucleotides of cytosine. Then coming to the nucleotides of guanine, GMP, GDP, GTP and cyclic GMP. Okay, nucleotides of guanine are guanosine monophosphate, guanosine diphosphate, GTP and cyclic GMP. GTP is required for protein biosynthesis, and if you remember the steps of TCA cycle, there the oxidation of succinyl-CoA to succinate, there GDP is phosphorylated to GTP. So that means nucleotides of guanine are involved in TCA cycle and GTP is required for protein synthesis. GTP and GDP both are the nucleotides of guanine. Then coming to cyclic GMP. Cyclic GMP already we have discussed, it is derived from the GTP by the enzyme. It is formed from the GTP by the enzyme guanylinate cyclase, membrane bound enzyme, and the activity of the guanylate cyclase is regulated by various factors like ANF, atrial natriuretic factor, enzyme guanylinate cyclase. The activity of the guanylate cyclase is being controlled by other factors like ANF, nitric oxide, sodium azide and nitroglycerin. So presence of all these factors stimulates the guanylate cyclase and increases the release of cyclic GMP. Increased cyclic GMP levels then involved in smooth muscle relaxation and vasodilation. So sodium azide, nitric oxide, nitroglycerin, they are powerful vasodilators. Why? Because they stimulate the enzyme guanylinate cyclase, thereby increasing the formation of the cyclic GMP from the GTP. Now this cyclic GMP which causes the relaxation of the smooth muscles, and it helps in vasodilation. Okay, that's it about the function of the cyclic GMP. Along with this, many coenzymes are nucleotides. Examples you can take NAD. NAD stands for nicotinamide adenine dinucleotide and its phosphorylated form. NADP, FAD, flavin adenine dinucleotide, coenzyme A. Okay, see here the nucleotides are the derivatives of the coenzymes. You can write the examples NAD, NADP, FAD, coenzyme A, etc. Okay, that's it about the physiological. This is the structure of cyclic GMP. That is the structure of the guanine. Guanine is attached to the ribosome. In the structure of ribose, both 3' and 5' has been esterified with the single phosphoric acid to form cyclic GMP. So that's it about the physiological importance of the nucleotides. So we have discussed adenine nucleotides, adenosine, guanosine nucleotides, its importance, and uracil nucleotides and guanine nucleotides with their physiological importance. So that means, what did you understand? So nucleotides, not only being the part of nucleic acids, they have other biochemical functions. Then coming to synthetic nucleotides, that I'll continue in the next class. Thank you.